the right protein in a living cell, and only those. That's the problem we tackle. This is BiGMINT. In drug discovery, a compound is tested against dozens of protein targets simultaneously. For each target, the question is binary. Does the compound activate or inhibit that protein at a given concentration? This gives us a table of activity labels, and it's almost entirely empty. In our dataset, less than 3% of entries have ground truth. So, the core challenge is, how do you predict compound activity reliably across many targets with almost no labels? There are two dominant ways to approach this. The first is chemoproteomics. You take the compound SMILE string and the protein amino acid sequence and model how they interact. This is target specific and interpretable, but it ignores what's actually happening inside the cell. The second is HCI. You treat cells with compounds, take fluorescent microscopic images, and read off the cellular response. This captures rich biology, but the image reflects everything the compound does at once, making it hard to isolate the effect on specific protein. Prior multimodal methods combine both, but with weak coupling that discards modality-specific information. BiGMINT addresses this with biologically guided hierarchical integration, using the molecular signal to focus the image and protein interaction prior to share information across tasks. BiGMINT builds the solution in several steps. First, for each protein target, we compute a molecular interaction embedding, taking the compound SMILES and the protein sequence and passing them through a physiochemically constrained graph neural network to capture how they interact. This gives us a target-specific anchor that carries protein identity through the rest of the network. Second, we encode the cell images. Each compound produces several image patches across multiple fields of use. We use a vision transformer pre-trained on disjoint large HCI dataset to extract patch features, which are then aggregated into a compact phenotypic representation, capturing the global cellular fingerprints of the compound. Here is the key insight. The cell images show the compound total effect. It's a composite of all the proteins being perturbed at once. So if we want to predict activity for a specific protein, we need to focus the image signal. That's exactly what our cross-model fusion does. It uses the molecular embedding, which knows exactly which protein we care about, to selectively amplify the parts of the phenotypic signal tied to that target. Think of it as using the molecular fingerprint as a lens to focus the microscopic image. The result is a target condition phenotypic representation, which we then fuse with the molecular signal for final activity prediction. Fourth, we scale this to multiple activity tasks at once using multiple task learning. Rather than training independent models for each task, we share representation across tasks. So a compound behavior on the one protein can inform predictions on related ones. Fifth, we bring in protein-protein interaction network as a biological prior. Proteins don't act in isolation, they interact in pathways. So task sharing related proteins should be able to help each other. We use the PPI graph to augment task embeddings, propagating signals between related tasks. This especially helps the hardest tasks which have the fewer labels. We evaluate on two large-scale datasets, U2S with 99K compounds and iNeuron with 40,000 compounds. Both have under 3% label density, closely reflecting real pharmaceutical screening conditions. The results are clear. On U2S, BiGMINT outperforms all baselines across every metric, achieving a mean AUC ROC of 78.2%, which is 10% gain over the best unimodal and 4.2 over the best multimodal method. But the more striking result is the task coverage. The number of tasks where AUC ROC exceeds 80%. BiGMINT covers 103% more such tasks than the best HCI baseline and 158% more than the chemoproteomics baseline. It doesn't just improve average performance, it unlocks tasks that were previously out of reach for every baseline. We also dig into why the two modalities help each other. We find that HCA is strongest for hub proteins, those with many protein-protein interaction, where the cellular morphological response is rich and detectable. Chemoproteomics shines for more isolated targets. BiGMINT outperforms both modalities across 54 out of 62 proteins, confirming it effectively harnesses complementary across full spectrum. Adding PPI prior further improved performance on 114 out of 170 tasks. Crucially, the biggest gains come on the lowest performing task, where borrowing signal from related protein makes the difference. The prior does not hurt strong tasks, it rescues the weak ones. BiGMINT shows that the right question is not which modality to use, it's how to let them inform each other, guided by biology. Chemoproteomics guided fusion, PPI prior, and multitask learning together set a new bar on compound activity prediction. We are looking forward to expand this to unseen proteins and richen biological priors. Thank you.